Imagine controlling gene activity with unprecedented precision, not by cutting or altering DNA itself, but by subtly rewriting its instruction manual. This is the promise of CRISPR-based epigenome editing, a rapidly advancing field at the forefront of genetics and molecular biology. This innovative approach, deeply rooted in the groundbreaking CRISPR technology, is rapidly evolving, with significant findings continuously pushing the boundaries of what's possible. Unlike conventional CRISPR, Cas9, which acts as molecular scissors to make permanent DNA alterations, epigenome editing offers a reversible and nuanced approach. It precisely controls gene expression by modifying epigenetic marks, the chemical tags on DNA and histones that dictate how genes are read. At its core, this technology leverages a catalytically inactive Cas9, or dCas9, which retains its ability to be guided to specific genomic locations. Crucially, dCas9 is fused to various epigenetic effector proteins, which are the true agents of change in this sophisticated molecular system. These effectors include enzymes like DNA methyltransferases, which add methyl groups to DNA, or histone acetyltransferases, which attach acetyl groups to histones. Conversely, other effectors act as demethylases or deacetylases, removing these tags and thereby reversing their regulatory effects on gene activity. These molecular modifications directly influence chromatin structure, the complex of DNA and proteins that forms chromosomes, altering its compaction and accessibility. By making regions of the genome more or less accessible to transcription machinery, epigenome editing can precisely fine-tune or entirely switch genes on or off. This locus-specific control over the regulatory landscape provides unparalleled precision without ever altering the underlying genetic code itself. A key advantage is its potential reversibility, meaning these epigenetic changes are not necessarily permanent, offering greater therapeutic flexibility. Recent advancements highlight the growing sophistication of this field, exemplified by a January 2026 eLife paper by Buttra, Cabrera, Spence et al. This study specifically addressed a major challenge: designing effective guide RNAs to reliably achieve desired levels of transcriptional modulation. The researchers demonstrated a method to accurately predict endogenous gene expression based on histone post-translational modifications, or PTMs, following dCas9-mediated perturbations. This groundbreaking approach significantly enhances machine learning models, improving the rational design of gRNAs for more predictable and precise epigenome editing outcomes. This technology matters significantly because it offers a safer and more flexible alternative to conventional gene editing. By avoiding permanent alterations to the DNA sequence, it mitigates serious risks like unintended mutations or chromosomal rearrangements that can arise from DNA cuts. Instead, it targets aberrant epigenetic states, which are often the underlying cause of disease, without touching the fundamental genetic blueprint. This approach holds immense promise for treating a wide array of diseases where gene expression is dysregulated. Conditions such as cancer, neurodegenerative disorders like Alzheimer's, and various immunological conditions could potentially benefit from this precise regulatory control. For instance, biotechnology companies are already advancing this concept into human clinical trials to silence the disease-causing DUX4 gene in facioscapulohumeral muscular dystrophy, or FSHD. Despite its immense potential, a significant caveat remains the challenge of efficient in vivo delivery of these complex dCas9 effector fusion proteins to target cells and tissues. While the eLife paper made strides, designing gRNAs to reliably achieve the desired precise levels of transcriptional or epigenetic modulation across diverse cellular contexts remains a major hurdle. Ensuring long-term stability of the desired epigenetic changes and rigorously investigating potential off-target epigenetic effects also necessitate continued research. As research progresses, these challenges are being systematically addressed by a global community of scientists and clinicians. Epigenome editing is not just an incremental improvement. It represents a paradigm shift in how we might approach genetic diseases. By offering a dynamic and potentially reversible control over gene expression, it opens up entirely new avenues for therapeutic intervention. How will this ability to fine-tune our genetic symphony without altering the score fundamentally reshape medicine and our understanding of life itself?